Hi, everyone. I'm Salma Karashi. Welcome to Neuroscientist Talk Shop, the University of Texas at San Antonio's Neuroscience Research Podcast. Today is April 8th, 2021, and I'm really pleased to welcome Alfredo Fontanini, who is professor and chair of the Department of Neurobiology and Behavior at Stony Brook University. He's also co-director of the Neuroscience Institute there. Hi, Alfredo.
Hello, Salma. Nice to meet you.
Nice to meet you too. Alfredo's work looks at how internal and cognitive states modulate neural responses to sensory stimuli in awake behaving rats and mice. Specifically, the lab studies how olfactory and gustatory circuits mediate the perception of taste, represent expectations, and control food consumption. His research relies on a wide combination of behavioral, computational, electrophysiological, and imaging approaches. So in the Zoom today, we've got an eclectic group that covers computation, theory, cellular, and circuit physiology, and taste. So we've got Fidel Santamaria. Hi, Fidel.
Hi, Salma. How are you?
We've got Lindsay McPherson. Hi, Lindsay.
Hi, Salma.
And we've got Charlie Wilson, as always. Hi, Charlie.
Hello.
I kind of thought a good place to start, a general place to start would the discussion would be on just cortical sensory processing in general. So the fundamental principles of sensory cortical physiology derive from classic work on the visual, mostly visual, but somatosensory and auditory cortices too. But it seems like many of the core ideas about things like topography and maps, cortical column structure, canonical circuitry, hierarchy, and integration of signal processing, they mostly don't apply to what we understand as the primary cortical areas for smell and taste. So can you say something about this disconnect? And if these modalities are really so exotic, taste and olfaction, should we be reverse engineering how we think about cortical sensory processing based on what's common rather than, thinking of these things as something so unique?
I mean, yes, this is a really interesting and very good starting point, I think. The gustatory cortex, and I think to some degree also the olfactory cortex, I've been studying mostly the taste cortex over the years, is quite unique on many accounts. From the architectural point of view, for instance, it's an area that is tucked between the somatosensory, which is 6 layers, and the olfactory, which is 3 layers. And it's a transitional area, which means that its structure changes from a neocortical template into a node called agranular template, meaning that it loses the classically defined layer 4. And this is just the beginning of its oddity. In a way, if you think about taste as a percept, it's unlike vision, for instance. It's different. It's difficult to abstract taste from pleasure and emotion. So I can show you an oriented bar, and all there is to that oriented bar, it's our orientation. This is not to say that visual stimuli are meaningless oriented bar, but that could be considered its basic building block. Whereas in the case of taste, you can take the taste equivalent of an oriented bar, a single molecule like sucrose, and you can dissolve sugar in water and you taste it. And sure enough, it has a physiochemical characteristic. It's sweet, but it's also palatable. It's also pleasurable. And so is, bitter is bitter and aversive. And so this intrinsic affective quality, this intrinsic value already sets the gustatory cortex somewhat apart from other sensory cortices, because you need to cope with that. It's very difficult to separate these affective and these physical properties. And so you could try to do it, or if you really try to fit the gustatory cortex in all the classic principles of sensory cortical function, then you end up with perhaps a lesser model. If instead you try to embrace all the quotes oddities of the gustatory cortex and study them as an integral part of perception, then you might actually find yourself with a very interesting model for studying how emotion and perception blend together into a unitary percept. And that's pretty much the approach that I've taken. I, like many others, have taken to the gustatory cortex over the past years. And so that allowed us to discover that, yes, it's an area where pleasure and sensation are integrated, thanks to connections, for instance, with the amygdala. It's an area where expectation plays a fundamental role in shaping the activity, again, thanks to connection with areas like the amygdala, medial prefrontal cortex. And so it's an area where reward, perception, and emotion really integrate. If you look at other cortical areas, it's not that they're actually not doing that. There's work, for instance, and there's more and more evidence coming in the past, I would say 5 to 10 years, that even the visual cortex, which is the champion of the most, the purest of most reductionistic of sensory areas, can represent a reward expectation, for instance, or can represent sensory motor expectation, movement. So it depends.
That's even true from primary visual areas, right?
Yes, exactly.
So it's probably just the speed of the signal, right? And how parallel the signal is, right? I mean, we have evolved, I mean, at least us primates have evolved to pay attention to the visual stimulus, and that is in parallel all the time. And it's around 120 hertz. I'm pretty sure sensory, gustatory, and you can probably calculate the speed of the sampling in gustatory and olfaction. You can do that. At least in olfaction, you can know how the little chaotic turbulence in the nose happens, and then you can figure out the amount of samples that you get, and it's probably not 120 hertz, and it's not as massive probably as.
So you're basically saying that since vision or audition are in a way faster than taste and olfaction, it's easier to, they seem face value more attuned at the physical properties of the stimulus. Whereas in the case of taste and olfaction being slower, they're allowed to reveal other properties.
I mean, that's an argument for olfaction, right? Olfaction is a way to preempt, right? And I mean, it's something that you're sensing from far away. And then you have time to decide if you're going to hunt it, like a polar bear is going to hunt a baby seal, right? I mean, they can smell them from 2 miles away or whatever, right? So that gives you a lot of time. Once you have the bear in front of you, you don't have that much time. You have to be fast.
I mean, another way in which the gustatory system is integrated with our functions, and I think like Lindsay here can tell you better than I can, is the integration with the interoceptive and with the homeostatic signals.
It sounds like almost what you're saying is that the time envelope of the action is what determines it, not the time envelope of the perception. So you wouldn't need to escape from a taste stimulus, right? I mean, can we think about it that way as opposed to?
And possibly like taste stimulus, there are only few exceptions where you need to have, particularly in experienced animals, there are only few exceptions in which you need to act really fast. And usually in our case is when your expectations are breached. Say that you're expecting to, I don't know, like a glass of Coke and it's root beer, and the sudden taste of root beer breaches your expectation. That may induce like a rejection response. Other than that, normally like the savoring happens on a fairly long time scale. It doesn't need to.
Olfaction though probably would, like smelling a predator would cause you to have to move very quickly and integrate everything really fast, I guess.
Yeah.
Hopefully if your smell's working well enough, you would have smelled it a little bit in advance, and so it would like Miguel was saying, you're smelling it from far away, so you have a little bit of time to like gather yourself and go and then make a decision, right? So I do find this temporal dynamic problem of the different sensory systems extremely interesting, and your work really kind of shows this. And even within that the epics of this responding is so interesting. And you mentioned too about like how, even as you're tasting something, one problem is just the physical nature of tastings, right? Yes, you're coating your mouth. They're going in all sorts of different areas to different times. It's not, I mean, as a peripheral taste person, I find this frustrating, because
it is messy. But that's why if you want to study the sensory coding from a purely psychophysical point of view, like we have such a wonderful control over visual and auditory stimuli. And whereas taste is slashing around the mouth in a somewhat uncontrolled way. So no, that's a very good point.
So do you think we're going to get to the point where we're no longer using taste stimuli and we just activate our taste receptor cells with some channel rhodopsin? Or are you going to try some of this? just get a hit of light on the tongue, and then get a few sweet taste receptor cells to fire, and then see what happens in the gustatory cortex.
I mean, from a point of view, like if one wants to have like a good control over the stimulus, that would probably be a good idea. I mean, I think in the olfactory system they actually have done quite some work in isolating inputs from single receptors onto specific glomeruli, and they have been using that to interrogate and to understand the coding of the olfactory system. In my case, I wouldn't know the degree to which this changes the dynamics, right? Maybe those sustained dynamics really need a protracted stimulation of the tongue. And so one would have to play around with those artificial stimuli. My way of controlling a little bit more has been moving from intraoral cannula to leaking. So there are basically two ways in which you can stimulate the gustatory system in rodents. One is through surgically implanted cannula that flush fluids directly into the mouth of the animal. Now, those are great if you want to study expectation, because you can really flush a stimulus that the animal has no clue about, neither the stimulus nor the timing in which you flush the stimulus. So in a way, those are a great way to hijack the animal's will. You couldn't do the same with leaking. But the problem is that you don't really know much of what goes on in the mouth. Maybe as far as we know, the animal might be moving the tongue internally differently for quinine and sucrose, and we wouldn't be able to see it. Whereas with leaking, at least you can monitor the leaking pattern, and with nowadays with decent speed cameras, you can also image the tongue from a couple of different perspectives and have a sense of the tongue's kinematic a little bit more. So I've been moving more toward that direction. The reality is that labs have compared the responses of the cortex to stimuli that are flashed onto the tongue or that are licked. And those basic dynamic blocks seem to stay the same. And so that seemed really to reflect a property of the system as a whole, more like forebrain interactions rather than peripheral dynamics. But I'm sure that there are also some peripheral dynamics that play a role.
Yeah, I was going to ask you about that too, because in your imaging studies where you look at the activity of the cortex, you're letting them lick. They're trained to lick. They need to be motivated by being somewhat thirsty to get them to do this. So would you think it would be much different? I mean, I guess from the electrophysiology, you would say that you don't expect to see much difference between mice that would be cannulated and forced to experience whatever taste you give them versus ones that are somewhat thirsty and are motivated to lick the taste.
Also the animals where we infuse taste with a cannula, they are thirsty, because we're basically delivering 10 microliters per 200 times, more or less. So there are a lot of fluids involved in the session. And so we need a cooperative animal in that sense. But I don't know. I don't expect them to have a huge impact. Obviously, we would not have the signals that precede the leaking. So one of the things that we are also observing is that every time the animal initiates a leak, there's some preparatory activity, very much like as if it was like a premotor cortex. Now, I'm not claiming that that's motor activity or premotor activity, but it's the signal is very similar. And so obviously that kind of activity is not there if you're passively flushing in the mouth of the animal. And I'm sure that there's also some segmenting of information similar to the sniffing in a way that happens with the leaking. But since 2-photon kind of like integrates all that, calcium imaging integrates all that over a long time course, I would expect the results to be pretty much similar. Fine dynamics may change on a very, very fine time scale, but not the progression through macroscopic, hundreds of milliseconds kind of epochs.
So some of that connects to the next thing I wanted to ask you about, which is about how your work in behaving animals has moved the needle on competing ideas about the spatial organization of taste responses in GC gustatory cortex that come from anesthetized rodents. So can you say something about that, and why is it important to be looking for topography, or is it still important to be looking for topography? What did you find, first of all?
Okay, so basically our work was motivated by conflicting evidence in basically in anesthetized animals. So in anesthetized mice and rat, it was shown by some groups that there was a very clear topography where basically there were clusters of neurons that would respond only to sweet, another to salty, another to bitter, and so on and so forth. Whereas other work showed some evidence of a little bit more overlapping representations. And we approached this, so in our electrophysiology, we never saw too much evidence of a clear topography. But then again, our electrophysiology was not really optimized to study the topography. So one of the problems of studying the insular cortex is that it's positioned in a way that is not amenable to mapping. It's difficult to reach the surface of the cortex. It's basically on the side. So unlike barrel cortex or auditory cortex, where you can actually, or the cerebellum, where you can go on the surface, do multiple penetration and map responses, this is much harder to do in awake animals. In the gustatory cortex. And so we never really saw any regional bias, meaning that with our multi-electrodes, we could record neurons that respond to sweet and to bitter, or neurons narrowly tuned to sweet and one narrowly tuned to bitter that were neighboring. So, but again, the electrophysiology was suggesting that there may not be a map in awake animals, but we really wanted to use the exact same technical approach and image on the surface using 2-photon or wide field and calcium imaging. And so we found that in alert animals, there's no real evidence of a gustotopic map, but rather more distributed representations. In a way, it's similar to what has been described in the olfactory cortex. Again, somewhat picking the chemical senses, taste and olfaction, apart from the classically neocortical areas like somatosensation, audition, and vision. Now, why that is important? Well, I think it's important. There are some practical aspects. So if there is a clear topography, okay, then it matters where you position your electrodes to study different phenomena. And so if it is true that the anterior part of the gustatory cortex is basically sweet cortex, then if you're interested in study, for instance, how sweet is represented, how the intensity of sweet is represented, then you'd better be in that spot of the cortex, right? Whereas if sweet is represented throughout the cortex, then you can be a little bit more agnostic on where you place your electrodes.
So are there kind of like psychometric curves that you can compare your detection of the individual taste sense and then compare that to the imaging work that you have done? I mean, I'm curious because, I mean, I go back to this thing of like the ecological importance of the function of that cortex. Is it about detecting like single pixels? Because we get influenced too much on this visual cortex stuff.
Yeah.
That it's about detecting the intensity, I mean, detecting one photon, right?
Yeah.
And then you have your photons, and then you generate your images and then your edges and stuff like that. But is it about detecting the actual concentration of the taste sense, or is it about the relationship of the taste sense with other taste sense? Right?
Yes. Yeah. No. The answer is, so these are all incredibly important problems and all very difficult to study, in the sense that your ability to sense the sensory space is really limited by how much fluid the animal is willing to drink. And so, if you want some statistical power in your comparison, you might want to have at least 10 trials, let's say, of each stimulus.
Right.
And each trial, each stimulation needs to be followed by some sort of rinsing. And that basically sums up that you could do, sure, you can do a psychometric curve but you can do it for one taste in one position.
Right.
So you can't really do it. You cannot do like the auditory system when you run these sweeps or you have these frequent beeps and you map all the space and then you do the experiment. By the time you have one psychometric curve for sodium concentration.
Because this should be important if we're going to make a comparison between the chemical senses and the rest of the senses. It's like, how come there's so much importance of the hedonic value?
Yes.
Right.
Yes.
And for example, in humans, the hedonic value reported, it's educational, right? I mean, an example is like chocolate. Mm-hmm. And I think it's the smell and also the taste. I mean, the tolerance for concentration of chocolate varies across the world.
Yeah.
Anyway.
No, but one might also argue, though, and some have, I think was a book of Mark Changizi, I think, on visual perception and the evolution of visual perception, that there's a great deal of attention that is devoted to elements like the oxygenation of the skin, whether someone is blushing or someone is getting pale, to the facial expressions, whether they're signaling calm, aggression. So one might argue that and that may also bring to some differences between primates and rodents, but one might argue that having such a wonderful control over stimulus of variables has kind of confined them to very reduced stimuli that has no affective content. While in reality, there's every reason to imagine that auditory cortices may be attuned to the affective qualities of the tone. If you're yelling at your dog, or if you're cuddling with your voice or your dog, these are very different meanings. So one might argue that also the other sensory systems have good reasons or good evolutionary reasons to be somewhat attuned to affective value. It's just that we never studied that way. Whereas with taste, since you can't parse it apart, But Fidel, I mean, I think the point of what's the function of the gustatory cortex is essential point, and we don't know. Like the answer is that we don't know. Like if you ask me personally, I think it's to integrate and to learn, right? We have a cortex that needs to process food. Food is an incredibly multidimensional construct where you have texture, temperature, olfaction, taste bundled together. All that is integrated on the physiological state of the subject and on the affective state. So to me, that's what the cortex does. It puts chemicals in the context of what goes on outside, in the mouth, and in the organism. But that is incredibly hard to study. We basically are kind of like bound to study only, you know, like let's say that this is like a three-dimensional picture. We kind of like study a couple of lines, like one line here to get a little bit.
I was doing a quick search on inkjet heads, printer heads, to see if you could insert one in the tongue and then just spray like and just spray. No, it says they're too big. You need a bigger rat or mouth.
You know, I feel I should apologize for doing this because you guys are talking about such important things, but I'm thinking about space again. So, and about that topography. So one of the reasons that topography in the sensory cortex is such a popular topic is because it integrates with our view of the circuitry. So there's short range connections, there are long range connections, excitation goes a certain distance, inhibition goes a certain distance. And so the idea is that those interactions are functionally important because of the topography. And that we can try to understand the structure of the cortex and the function of the cortex all in one go. So, and you were just saying that the structure of the gustatory and also the olfactory cortex is a little different from the neocortical regions that are the other ones. And then the other example I was thinking of is the olfactory bulb itself, which does not really have a spatial topography, but which has this wonderful glomerulus skipping long distance inhibition that brings all these faraway glomeruli together functionally. So can we interpret the gustatory cortex that way? Are there features of the interconnections of the cortex? What is the structure of the cortex?
So this is a very interesting point. In some of the modeling work that we have done. So the gustatory cortex, when you record, for instance, the spontaneous activity of the gustatory cortex, one could define the spontaneous activity as metastable, meaning that you have some patterns of activity that remain constant for a few hundreds of milliseconds up to a few seconds. And then all of a sudden, the whole ensemble changes its activity coherently, okay? And so we found that we can reproduce this kind of metastable activity in a network that have a specific architecture, and it's a clustered architecture. And a cluster architecture is basically a network where you have ensembles. You have groups of neurons that are more strongly connected with each other than across these groups of neurons.
But not on the basis of space.
Pardon?
Not spatially, but just
not spatially. So the model is agnostic, right? It could be spatial, it could be hotspots, or it could be far apart. And to be totally honest, my motivation to get into calcium imaging was to be able to visualize in a spatial way these ensembles and whether they were distributed or concentrated. That was one of my motivations. And now it turns out that for many accounts, calcium imaging, so the whole theory that we have with metastability very much relies on the speed of spiking, and that we lose resolution when we look at calcium imaging. But the issue remains the same, that whether we can experimentally measure these ensembles of neurons. Now, there are multiple questions, right? One is whether these ensembles need to be ensembles of neurons that respond to the same features. And on the one hand, it's tempting to think that, oh, if you have neurons that are narrowly tuned to sucrose, they should have some sort of preferential connectivity among them. But part of the discussion that we had so far, and also part of the imaging work that both from Zucker's group all the way to ours, is that whenever we give taste, there's so many things that so many responses that we don't see, that makes you wonder whether the ensembles are attuned at either meta properties or other properties of taste. But so practically speaking, getting into a little bit more the nuts and bolts of the cortical connectivity, the lab of Arianna Maffei has done some connectivity studies with paired recordings. So what they typically do, and they have done this in the past in the visual cortex, and now they're doing it in the gustatory cortex, they land basically 4 patch pipettes, and they patch onto quadruplets of neurons in search of connected pairs. And then if they don't find a pair, they move to the next quadruplet, and so on and so forth. This is a relatively higher throughput than just like doing a pair. And it seems that with regard to pyramidal cells, the connectivity in the insular cortex, of course, like this approach is biased by cells that are neighboring, right? And so the connectivity in the gustatory cortex of neighboring cells is much lower than in classical neocortex, in V1, for instance. This seems to suggest that the connectivity might be more of a medium range and medium distance. So what we're hoping to do soon, we're trying together, both labs, to set up one of those systems where you can do an optogenetically assisted circuit mapping, where you kind of like give patterned light stimulation and focal stimulation. And we're hoping to get at that question with this approach. I also seem to recall—
You should come and do the experiments with our new microscope.
Oh, I should actually.
You should.
Happy to come, anytime COVID lets us do.
Yeah.
So one of the things, I also seem to recall that in the piriform cortex, which has a beautiful, beautiful organization, very logical and very geometrical, while you have a lot of recurrent connections, they're mostly from medium range distance. So in fact, when you record from neighboring pyramidal cells in the piriform cortex, you don't necessarily have as high of a connectivity as in the neocortex. So maybe like this distributed organization, these lacks of map, these sparse representations are also associated with this like more like medium long range reciprocal connectivity, whereas in S1, in the barrel cortex, you have everything that is more packed closely together.
And do you think that relates with that ability that you saw to make these very plastic associations, like when you were trying all these different stimuli to see if they coupled with, or they could couple with anticipating a taste response, right? And you saw like at baseline level, there was a few neurons here and there that would respond to a light cue or to an olfactory, or I forgot to get the auditory cue, a few more that would have a somatosensory kind of phenotype and whatever. But then after you had done the paradigm, right?
Yeah, they
training them. Then they were like, bam, like they were just like all of these neurons were responding to this cue. So do you think that organization would kind of improve that ability or in some way?
So I think probably it's, so if you have an area where neurons are narrowly tuned and reciprocally connected, in a way, you're hardwiring the salient features of the sensory space. If instead you have an area that is a little bit more wishy-washy, in a way where you have less strict columnar organization, connections that are a little bit more spurious, where you have like a sweet neuron that could be connected with one that does odor. And so you have neurons that encode all these features. Then it's easier. You have basically a neuron that is synaptically more responsive. And so then it's easier to do some pairings where a stimulus that before being paired was below threshold, then it becomes above threshold. One other thing that we're also seeing is that the overlap, so the neurons that respond to multiple stimuli seem to be like this breadth of tuning in a way, this broad connectivity may be important for animals to even be able to perform a task where you take two sweets and you ask them to separate those two sweets. So in the seminar, I presented some experiments in which an animal had to sample some tastes from a central spout and then associate with the taste, with a reward delivered at two lateral spouts. And we trained the animals that one sweet or one type of bitter would lead to reward at a left spout, whereas another sweet and another bitter would lead to reward to a right spout. So this task, in a way, requires some sort of, let's see if I can say, orthogonalization of the sensory space, where you go from encoding sweets and bitters together to encoding stimuli that predict the direction of the reward. And so for a network to be able to do that, you cannot rely on ensembles that are very well separated. In fact, with the model that we had developed of the cluster network that we developed with in the gustatory cortex, some of the preliminary data suggest that if we want that model to reproduce the spiking activity that we see in this decision-making task, this odd decision-making task, we need ensembles to not to be narrowly tuned, but to be overlapping, so that you can form those plastic representation where at the beginning you pay attention to sweet, and then you pay attention to whether sweet or bitter indicates going left or going right. So that, like naively, that's one of the functions that I would think some sort of mixed tuning or like networks that have a fairly high reciprocal connectivity could mediate. Whether that needs to be packed in the same space or spatially distributed, that I don't necessarily know. But yeah, so we these are I think incredibly important topics of general cortical function. And what we tend to do is, as we keep finding these exceptions, we ask ourselves whether we really came across an oddball system, or whether, you know, in a way, these are, every sensory cortex can be seen from different perspectives. And maybe our taste perspective, our perspective on the gustatory cortex can help us looking at some hidden features of somatosensory or visual cortex, for instance. So that worked for us as a source of inspiration. I never had the guts of testing the hypothesis in the visual cortex, but fortunately there are other people who are doing it for me.
So I'm trying to, we're off the usual time script here. So we have until 4:30, is that right? So we have another 15 minutes. So I'm gonna cut this part out. But do we want to bring up anything new? I actually wanted to talk about the discrimination study, some of the questions that came up at the end. I'm not sure if we have the time to really draw that out since we have such a little bit of time. I could sort of give you a last word right now also, just what you kind of just did, but what do you guys think? Do we want to take it another 15 minutes? Okay.
I have time.
Okay, good. So I wanted to ask you about that discrimination study that you just mentioned. So in that work, you you show this evolving response of sensory to motor. And one of the really interesting parts about that I heard was that, and you can correct me if I'm wrong, that early phase where you have the discrimination task happening, where you have a sensory coding of the response in the gustatory cortex, that you were able to see the discrimination in those neurons. But then when those neurons were taken offline, the outcome of the task wasn't affected. So
yes.
It wasn't performing, the cortex, these neurons were not actually performing the discrimination that led to the correct response in the task. Whereas, I mean, that just seemed like a very surprising result.
No, yes, it was very, very surprising to us. So basically, we use optogenetics to either silence the activity in the cortex during the period in which the animal is sampling the taste, or silence the activity in the cortex in the delay period during which the animal is deciding whether to lick left or to lick right. We find that silencing during the delay has an impact on the performance. The animal makes more mistakes. And whereas the silencing during the sampling has no impact. And I think like this really is surprising, and was one of the questions that, and it's perhaps one of the questions that comes up more often, and could be interpreted in all sorts of different ways. We are a bit agnostic about the interpretation. So there could be some very technical reasons that, for instance, the activity, there's more taste activity. The taste activity in the cortex is more robust to silencing than the decision-making related activity. So maybe there's the elevation in firing rates is less sensitive to silencing than in the case of the decision-making activity. So this could be a technical explanation. Another explanation could be that once you train an animal for, in the case of that task, that was anywhere between say 6 to 8 weeks to perform a task, at some point, you no longer need the cortex. You know, much like striatal circuits in habit learning, you know, they shift the emphasis on different portions. One could argue that could happen something like that also in the gustatory cortex. Or the gustatory cortex is not needed for this task. I have no problem believing that the gustatory cortex is not needed for simple discriminations. For these tasks that seems requiring this orthogonalization that is like, you know, against the innate organization of taste, I would be inclined to think that that's such a difficult task that may require the cortex. Another possibility, again, is that you have taste signals in the gustatory system that remain beyond the silencing. So the thalamus, for instance, produces responses that last for about 2-3 seconds. So if you're silencing the cortex, you might be taking the cortex offline for that half a second following taste stimulation. But then when the cortex goes back online, it's receiving information from the thalamus that allows the cortex to kind of like catch up and perform nevertheless. So these are some of the possibilities. We are very interested in understanding what, if any, the gustatory cortex role plays in taste and just in taste discrimination. And so now we have a mixture detection task. We're basically training mice to on a very simple task where they need to lick left for sweet, lick right for salt. And then we test these mice on a series of mixtures where there's 90% sweet and 10% bitter, 10% salt, up to like 55% sweet and 45% salt, and vice versa. And this conceivably has some concentrations that are very hard to discriminate for the animal. And we're looking into silencing the gustatory cortex at different times in these tasks to see whether the psychometric curve gets modified by the silencing. So maybe this is also a task. We can train mice on discriminating sweet and salt in about a week. So we're also kind of like shrinking the time. And the concentrations, they're not trained. You're just using the concentration to probe the perceptual space of the animal. So maybe this is one of the situations where we could see an alteration in the activity. If we don't, then we will conclude that the gustatory cortex is not there to detect taste, or that animals can detect taste without the gustatory cortex. Whether that means without the conscious perception of taste, who knows?
Excellent. Thank you for joining us. And this, it's always so relevant to so many of our own, I was listening to your talk today, I was sort of thinking about conditioned taste aversion, for example. It's such a powerful thing, and we all have some reference point to something that we tasted that we just cannot eat anymore. Charlie and I were talking about this earlier. Enchiladas are the no-go for Charlie ever.
Oh,
sorry. So, but it's an incredibly interesting combination of models, and just thinking about this idea of primary sensory cortices. Yeah, that was sort of what I wanted to get a sense from you, is that are we thinking about this in terms of this sort of hierarchical coding of taste? It really doesn't apply to taste, this idea of building perceptions and integrating, and it's really serious.
So there was a moment in which the idea of a hierarchical coding was adopted in chemosensation. For instance, for a relatively long time, researchers thought that the perception of flavor, the integration between taste and olfaction, would happen only in the orbitofrontal cortex, with the idea being that gustatory system does taste, olfactory system does olfaction, they come together in the orbitofrontal cortex, and that's where you have flavor. But more and more evidence is now pointing at the fact that it's really distributed, and that certainly for the case of taste, since eating is such an integrative experience, it makes sense to have an area that is actually integrating instead of separating. The idea came every now and then, and comes and goes, that maybe the different divisions of the gustatory cortex, the granular, dysgranular, and agranular, can correspond to different levels of hierarchy. We never found in our recordings backing to that, meaning that we can have anticipatory responses dorsally in the granular, or you can have narrowly tuned neurons ventrally in the agranular. So in our case, it seems that there's a lot of overlap of functions. So yeah, so much of our work challenges the idea, or much of the work on the, not just ours, of many people on the gustatory cortex challenges this idea of primary, secondary, basic feature detection, and then assembling of a more complex and abstract representations.
Thank you for joining us. Great. We really appreciate the time. Thank you, Fidel, Lindsay, Charlie, and of course you, Alfredo Fontanini.
Thank you so much. This was lots of fun.
Thank you, Alfredo.
Inviting me.
Scientist Talk Shop, guys. Thank you.
Take care. Bye-bye.
Wait, don't.
Alfredo, you
Yes.
almost said the gustatory cortex is there to substitute for its own absence. That's what Jim used to say about the cerebellum. They were this close.
Well, you can tell. Well, yes, I think you can tell a lot of you can see the lineage, right?
Yeah. You were about to say it. I think I'll give you another 5 years, and you will start pontificating.
And then I'm ready to write a review, taste for motor sake or motor.
You've been thinking about it.
You've got a draft. You've got a draft.
Yeah. I mean, just I'm working on it.
Yeah. Well, thanks, guys.